Hi! Today in this video, we will talk about the Calvin cycle in photosynthesis. So, what is Calvin cycle? Calvin cycle, also called as light-independent reaction or dark reaction. This cycle builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH. Calvin cycle has three phases: carbon fixation, which is catalyzed by Rubisco, reduction, and regeneration of the CO2 acceptor or BP. What's gonna happen in the Calvin cycle is first, carbon dioxide will enter the cycle. It is going to react with ribulose biphosphate catalyzed by the enzyme Rubisco. And that's gonna turn into 3-phosphoglycerate represented by PGA. And that's the carbon fixation, the first phase of Calvin cycle. Where BP is a molecule with 5 carbon atoms, where in carbon 1 and 5, there are 1 each phosphate group. So, there's 3 RuBP that is going to react with 3 carbon dioxide molecules. Now 1 molecule of carbon dioxide reacts with 1 molecule of RuBP. That's why we're going to get 6 carbon molecules, but it's going to be broken up into 2 3-carbon molecules. So, PGA is a 3-carbon molecule with 1 phosphate group. Now, to keep track the total number of carbons, if each RuBP has 5 carbons, 3 of them has 15 carbons plus the 3 carbons from 3 CO2 molecules. So now we have a total number of 18 carbons. So, thus, there's 6 3-phosphoglycerate molecules. Now, the enzyme kinase is an enzyme used to transfer a phosphate group to a molecule. So, here, it will going to require ATP. We're going to use 6 ATP molecules to phosphorylate 3-phosphoglycerate, and now we have 1,3-biphosphoglycerate. We have still 3 carbon molecules, but now we have a phosphate group on carbon 1 and carbon 3. And with that, we can see that PGA kinase is an enzyme that catalyzes the conversion of 3-phosphoglycerate into 1,3-biphosphoglycerate. So, ATP gives up a phosphate to become ADP, and that phosphate is transferred to this molecule. And as you can see, we now have 2 phosphate groups. So anytime you see a kinase enzyme, it catalyzes the transfer of a phosphate group from one molecule to another. Now, the next step is to use 6-NADPH to reduce 1,3-biphosphoglycerate into G3P glyceraldehyde 3-phosphate. So 6-NADPH is going to convert into 6-NADP+. This reaction will be catalyzed by the G3P dehydrogenase enzyme. This enzyme, from the word hydrogenase, it removes hydrogen from NADPH. At this point, we're going to get 6 G3P molecules. One of 6 G3P molecules is going to use to produce sugars like glucose and fructose. And the other 5 G3P molecules is used to regenerate the 3 ribulose biphosphate molecules. So, remember that the total number of carbons here is 15, and that each G3P molecule has 3 carbon atoms with the phosphate group on carbon 3. So 5 times 3 will give us a total of 15 carbons, which is what we had in the start. Notice that the net result of the Calvin cycle is converting 3 molecules of CO2 into 1 molecule of G3P. Now, Calvin cycle is how plants normally function. But what if there is no water available at the moment? What will going to happen? Well, there are alternative mechanisms of carbon fixation that have evolved in hot, arid climates. Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially photosynthesis. On hot, dry days, plants close stomata, which conserves H2O but also limits photosynthesis. The closing of stomata reduces access to CO2 and causes O2 to build up. These conditions favor an apparently wasteful process called photorespiration. So what is photorespiration? Photorespiration limits damaging products of light reactions that build up in the absence of the Calvin cycle. Photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more carbon dioxide. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. So now let us discuss the three types of plants: the C3 plants, C4 plants, and CAM plants. C3 plants. In most plants, C3 plants' initial fixation of carbon dioxide via Rubisco forms a 3-carbon compound, 3-phosphoglycerate. In dry environments, the stomata close to reduce water loss and blocks oxygen that causes to build up in the plant. The plants that utilizes this process include soybeans, oats, wheat, and rice. In photorespiration, Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle, producing a 2-carbon compound. Photorespiration consumes oxygen and organic fuel and releases carbon dioxide without producing ATP or sugar. C4 plants. C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into 4-carbon compounds in mesophyll cells. The plants that utilizes this process includes corn and sugarcane. C4 will close stomata to conserve water loss. However, unlike C3 plants, these plants will continue to make sugars using photosynthesis instead of entering photorespiration. CAM plants. Some plants, including succulents, use crussylation acid metabolism or CAM to fix carbon. CAM plants open their stomata at night, incorporating carbon dioxide into organic acids. Stomata close during the day, and carbon dioxide is released from organic acids and used in the Calvin cycle. The plants that utilizes this process includes pineapple and cactus. So that's the Calvin cycle and a brief discussion about photorespiration. Thank you for watching!